Yes, hello everyone. I'm decided to start 6 minutes before 4 o'clock my time here. I'm here with Sy Garte. Hello, Sy.
Hello.
Nice to have you at my channel first time, and it's a pleasure to have you here and to talk a little bit about abiogenesis, evolution, origins. I know that you are a Christian and evolutionary biologist, and you're a theistic evolutionist. Is that correct?
Yeah, more or less.
Okay. So before I went live, you started to telling me about your recently published paper. Maybe you can go on and tell a little bit about it.
Sure. So what happened, you asked me what my research fields are, and I mentioned my research fields when I was an active professor doing research. But then once I retired, I began pursuing an interest I have in, and I've had for a while, in the whole issue of origin of life. And a lot of people, of course, are doing research in that field, but my interest is very specific, and it has to do with the question of self-replication and the accuracy of self-replication in life. And that's what this paper is about. It was recently accepted for publication in Acta Biotheoretica, which is a mainstream peer-reviewed scientific journal. There's nothing in the paper that says anything about miracles or God or anything like that, of course, because it would never have been published. But the paper does make some points that I think are important in any discussion of the origin of life, including the concepts of intelligent design, evolution, and the role of what still we consider mystery about how life began. So that paper is currently, it's been accepted. It's available online. I will send you a link so that people who want to can look at it.
Please.
In the description. And I, if anyone is really interested, they can easily find me on Twitter, just Sy Garte, or on Facebook, and they can just ask me, and I'd be able to get them a PDF fairly easily. So I think the paper will eventually, well, it's now, as I said, it's available online. It will be published in print form probably in a few months.
Okay. So you said that your focus on that paper is about self-replication.
Yes.
And do you actually believe that life can come from non-life without God's intervention, that it can originate by natural means, by self-organization? Or do you believe that God created life and then let life to diversify by itself through evolution? What is your view on that?
Well, I don't have very rigid views. Some of my views have changed a little bit with time. They keep changing, which is actually fairly common in science. But I would say that, first of all, I believe God created everything. The first verses of Genesis make it very clear God created the heavens and the earth, and that includes everything. Now, the details, where the devil lives, of course, I'm not sure. I could go, if somebody figured out that there is a path to the origin of life through purely natural chemical processes, I could be convinced, but I frankly don't think that's going to happen, because if it were going to happen, it would have happened already. There are some fantastic labs doing research, and the number of problems with an abiogenesis from chemistry to life is just staggering, and it's as James Tour often says, the media doesn't cover it, the mainstream journals don't cover it, but just the chemistry part of it is extremely difficult to imagine. And I'm focused a little bit past that. I'm focused on what happens. Let's say everything works out great and you have some self-organization and you actually end up with a cell. Well, then what? The cell can't evolve. Because evolution, and this is the part that I really stress. When people talk about evolution, they talk about natural selection and mutations. But there's another aspect to evolution, which is almost always just skipped over and taken for granted, and that is the question of accurate, very, very accurate self-replication. Remember, nothing in the world self-replicates. Nothing. The only thing that self-replicates are living cells. Not even chemicals self-replicate. DNA does not self-replicate. RNA does not self-replicate. Crystals, no, they don't self-replicate. They grow. And same with storms, all kinds of things. There's nothing in the physical world outside of a living organism that self-replicates. And this, I think, is an extremely important point that's often overlooked.
Yes, I make an analogy, which I call the factory maker argument, where I say that cells are chemical factories. And when I bring up that argument, then a very common objection is, well, but factories do not self-replicate. And my answer to that, of course, is that is actually evidence that we cannot make factories which self-replicate, because that would be such a complex thing that, cells, they are working entirely autonomously. They do not require any input or intelligent input or so. So they are working entirely autonomously. So that actually adds a huge amount of argumentation for intelligent design.
Exactly.
And not against it.
Yeah, the story I like to say is, let's say you have a house and a couple is living in a house. They love the house. They have a couple of kids and it's in a beautiful neighborhood. It's a great house. Everything they have in it is great. And the kids decide they want a house exactly like that. And so they tell their parents, I want a house right next door, right near you, but exactly identical to everything in this house, including the furniture, the paintings, the books, the clothing, everything the same. Do that. And you could do that. You could hire, let's say the parents say, okay, we'll hire a contractor, we'll get an architect, we'll look for all the same furniture, we'll buy all the same books if we can find them, And you could do that. It would take a long time and a lot of effort. But one thing you cannot do is find a house that replicates itself. And a house, no matter how much you have in that house, no matter how much you have stored in the closet or in the attic, contains nothing compared to what's in a cell. A cell has 1 billion protein molecules. And and that's just the beginning. All the organelles, all the membranes. And if you look at a cell that replicates, let's say a simple bacterium, and you look at the two daughter cells, they're pretty much identical to the original parent cell. And that happens every 20 minutes.
Yeah, so when I investigated the number of proteins in the cell, I was really astonished when I did read that the human cell has 2.3 billion proteins.
That's right.
And I cannot even imagine such a quantity of proteins in a single cell. That's just staggering.
That's right. It is. And a bacteria has 1 billion. So humans are about twice as many proteins, which is about right.
But I don't know what kind of bacteria that has 1 billion proteins. But I usually always make the argument that the smallest life form is not a mycoplasma, which is commonly used as an example with 500,000 nucleotides, but it is not free living. It requires a human host. So it has to import all the amino acids in order to to survive. So I say mycoplasma isn't a good example as a minimal free living life form. And the one which I usually use, which is one of the smallest, is Pelagibacter ubique. And it has 1.3 million nucleotides, so about 2.5 the size of mycoplasma. It's considerably bigger. Yeah. So in order to have an origin of a genome with 1.3 million nucleotides, the odds are 10 to the 722,000.
So, yeah, I mean, you know, when I have made this argument, and I've made it to friends of mine who are origin of life researchers, a couple of whom are Christians as well, and I've made this argument about, you know, the origin of replication. How did you get that? And because, you know, if you hold to evolution, you tend to think that everything that modern life has evolved because it was better. So there was natural selection and it evolved. So the first answer you get is, well, okay, well, obviously replication is extremely beneficial because a cell that can replicate is immortal in a sense because it keeps replicating, whereas a cell that doesn't will go away, and that's the end of it. But then my question is, but wait a minute, evolution to work in order to allow these things to develop has to be already include replication because the whole point of evolution is it's not, you know, if a bird gets a better eye and has a better eyesight, that gives him a selective advantage. But if his kids don't inherit that, what good is it? The bird has his claim to fame and, you know, sees stuff, but the kids don't inherit it, so there's no evolution. So you have to have accurate inheritance even to start evolution. So how do you get accurate inheritance before those accurate inheritance?
Yeah, I am frequently at another channel of speed, gravity of sound, and there is a biochemist. His name is Dimitar Kunev, and he also publishes science papers, and we had a few interactions. And his claim is that evolution actually can start prior to DNA replication and when life starts. What would your answer be to that claim and assertion?
I'd like to know how, because if you don't have any replication of a genetic material, maybe you have RNA replication, but that's a big problem anyway. But you have to have something replicating. Otherwise, I don't know how evolution starts without that. I can't imagine it. Maybe he has an answer. I've seen his papers. I haven't read them, and I haven't read that one in particular. But I don't see any mechanism. If you don't have, you know, you could have a very good cell. Okay, you could have a great membrane with all kinds of enzymes inside that are metabolizing away. You could have an energy generating system, beautiful, great. That cell may live for a long time. It may have a very high probability of survival, but eventually it's going to die, and then you're done. I mean, if it doesn't replicate itself, so how do you get evolution there? I don't get it. So yeah, go ahead.
So the proposition of theistic evolution, is it that God simply created the chemical elements and left them by their own, and then life also started by chemical evolution? Or is the proposition that God actually created life and then eventually a single life form, and then it diversified, and then evolution kicked in? What is there a consensus about the proposition of theistic evolution, or has each one a different understanding of this?
Yeah, if you go to the BioLogos website, for example, which is the main home of evolutionary creation, which is trying to replace theistic evolution, but it's the same thing. But if you go to the BioLogos website and you look for that, what you'll find is that they say, and they're correct, that there's a wide range of views. There are people. There was a paper recently in Proceedings of the in Perspectives in Science and Christian Faith, PSCF, which is the journal of the American Scientific Affiliation, ASA, which I'm a member of, I'm a fellow of, and I've published in that paper. And just parenthetically, I'm also the editor-in-chief of God and Nature, which is a magazine that the ASA puts out. But getting back to what to this question you ask, that paper, which was co-authored by Jim Stump, who's the vice president of BioLogos, put forth the idea that God created the universe, and pretty much after that, natural laws took over, and the natural laws include the laws which allow life to emerge from chemistry, but no details were given. And I don't hold that view, it's not necessary. In other words, you don't have to hold that view to call yourself an evolutionary creationist, because frankly,
sorry, give me just a minute, because
sure.
I have to open the door to my employee. Okay, just 2 minutes, please.
Mm-hmm.
Okay, I'm sorry.
Okay, no problem.
Yeah.
So what I was about to say is that very quickly that what I was about to say is that the key thing to understand here is that abiogenesis is not considered to be part of the evolutionary story. And that's important, because I think it's a tacit admission that we know the mechanism of evolution, but it can't be, at least I believe, it cannot be the same mechanism that would explain the origin of life. And even Darwin, didn't talk about it. Richard Dawkins says very little. He just says it only had to happen once, and then he says a few other things. But in his better books, he says that's not something that is part of evolution. And so you can be an evolutionary creationist, meaning that you believe that, God, that evolution is one of the laws of God, basically, like gravity or electromagnetism, but that doesn't mean that you're committed to a natural cause for abiogenesis.
Okay, so you would agree in that case that probably God is a better explanation for the origin of life. Is that correct?
Correct.
So do you believe that the common ancestry is true?
Oh, yes.
Now, I have investigated that, and we know that the replication mechanism of prokaryotes and eukaryotes is quite dissimilar. It is different. And there are other points as well, like the membrane of prokaryotes is different than eukaryotes. Sure. And also the ribosome is different. And also the carbon fixation mechanism is, they proposed the reverse TCA cycle and so forth. But we know that there are 6 different carbon fixation mechanisms in life, and there are several difficulties with that view. How do you deal with that?
Well, you're right. The prokaryotes, eukaryotes, and archaea as well have differed considerably in the details of all of those mechanisms. But on the other hand, all living organisms, no matter what kingdom they're in, have the same genetic code with, again, some small differences. That's true. There are a few organisms with a slight difference in the genetic code, but they're off on a branch. So the vast majority of life uses the same genetic code and uses the same general mechanism for replication of DNA, for synthesis of proteins, which is an incredibly complex mechanism that requires many, many steps. And that's the mechanism that I don't see how could possibly have evolved, even though it's so incredibly complex. That's where actual irreducible complexity really is true. And this has been stated not just by me, but even by atheist evolutionists like Eugene Koonin. So I think that the fact that there are differences is correct, but the commonality of the basic mechanisms of life can be traced all the way back to what we call Luca, the last universal common ancestor.
Yeah, but for example, I have listed 37 different genetic codes, in special mitochondrial codes, genetic codes. There are quite a number of different variations. And if the endosymbiotic theory would be true, and we know that mitochondrial DNA is rather small, it has about 20,000 nucleotides.
Right.
And if we suppose that there was an endosymbiotic event where one organism engulfed another one, and if we suppose that these organisms had a minimal genome of, as I stated before, about 1.3 million nucleotides, then there would have preceded a huge devolutionary event where you go from millions of nucleotides to only 20,000. And then you would have also to change from the universal genetic code to the extant now mitochondrial code, which is different. So would that not be a huge difficulty to have that transition of devolution and change of the genetic code in order to have that endosymbiotic event?
Yeah, I'm not quite sure I'm following well that, but so in other words, what you're saying is that the mitochondria, who were originally a cyanobacterium, have a smaller genome than the current eukaryotes or even current prokaryotes, right?
We can take a smallest free living bacteria, which has about 1.3 million nucleotides. Okay, now let's suppose
that's a modern bacterium.
No, it is a modern bacterium, but it is one of the smallest that we know that if we reduce it even further, then essential proteins will not be there.
Right.
Okay, so if we take that as a model and say there was that endosymbiotic event where one organism engulfed the other one, then you would have to have that devolution from 1.3 million nucleotides to about 20,000, which today are used in mitochondria. that would be a huge evolutionary step.
Well, I think there's a problem there. It's clear. I mean, first of all, you have to think about this event occurring in the far distant past. We don't know what the size of the genome of the engulfing organism was. It might have been somewhat lower. And we don't know what the size of the cyanobacterium genome was. It was probably considerably higher. What happened once mitochondria entered cells is they lost a great deal of DNA, which is not needed. So we can't say that there were 20,000 nucleotides in the original bacterium that was engulfed. There may have been quite a bit more. It's also possible that it was a small. Obviously, it was smaller than the engulfing cell, so it might have had fewer nucleotides. The modern picture of all bacteria is not probably very close. Remember, bacteria have been on the planet for just about 3.5 to 4 billion years. Longer than anything else. And so during that time, as many people have shown, there's been a gigantic amount of evolution in bacteria. And modern bacteria are not simple organisms. They're incredibly complex. They have they speak with signals. I mean, there's all kinds of chemical stuff going on with bacteria that is astounding. And it's not surprising because they've been around for so long. So I don't see, and the other thing I'm not clear on is the difference in the mitochondrial genetic code to the genomic genetic code. Could you elaborate on what those differences are? I'm not familiar with.
Well, I have a link here. I'm not, since I did collect that information a few years back, I cannot give you details in regards of that, but I can put here the link, and then after our conversation, you can have a look on these codes. And then we can eventually have a follow-up and elaborate on that. Give me just a minute here until I find it here.
Oh, yeah, that's right. There are some differences. You're right. Now I'm remembering. Okay, it's been a while since I worked on mitochondrial DNA.
Yeah.
There are some small differences. They're very small, though. They're not major.
Let me just find it here. Okay, the different genetic code. I'll put here the link so everyone can have a look at it.
Yeah, I'll take a look. I do remember small differences now.
I never put it here also in private chat, so yeah, afterwards you can look it up.
Okay.
But I can quickly mention here a few. Maybe you remember if I mention them. Yeah. Actually, it's 31. The standard code, the vertebrate mitochondrial code, the yeast mitochondrial code, the mold protozoan and co-elentrate mitochondrial code, and mycoplasma, spiroplasma code, the invertebrate mitochondrial code, the ciliate hexametanuclear code, the echinoderm and flatworm mitochondrial code, the euplotide nuclear code, the bacterial, archaeal, and plant plastid code, the alternative yeast nuclear code, and so forth. In total, I'm listing 31 different codes.
Yeah, I have to look at that. I think there might be an issue there.
And there is another point, Sy, which is that we only know of eukaryotes which have the ability to engulf other cells, but we don't have any example of prokaryotes being able to do this. And that would be, in my understanding, as well be a problem, no?
I'm sorry, I was just looking at that quickly. Let's see. I don't know. Remember that the endobiogenesis concept is a proposal originally made by Lynn Margulis, if I'm not mistaken.
Yes. Yes.
And it has some evidence, but it doesn't, it's not something that can be proven since it happened only a couple of times. And I don't know what the alternative is. I guess the alternative, I mean, what would be the alternative to endobiogenesis?
I mean, I think that the transition from prokaryotes to eukaryotes is one of the biggest evolutionary gaps. And I mean, that would be something that would have to be solved if someone wants to claim that common ancestry is true. And there are actually even science papers which openly admit that the tree of life, starting with eukaryotes and archaea, would be different than bacterial origin. So
right. So let me talk about that. That's an interesting point. The point about, you're right that the prokaryote to eukaryote transition is very unusual in evolutionary terms because it didn't happen slowly and gradually. It happened in one big step. And that was the revolutionary idea that Lynn Margulis proposed, because remember that Darwin originally had said that gradualism was critical to his theory. Yeah. That every change has to, he said that if that could be disproven, his theory would be wrong, which is that every change would have to be itself small so that it could happen and have an advantage, a selective advantage. That's been disproven. One of the things that goes against that, and by the way, that's called the continuity principle. And the reason that's been disproven is because the evidence, which includes the engulfing of one cell by another cell, and it also includes, for example, some of the very rapid evolutionary changes that occurred, that we know occurred, based on things like, the Burgess Shale and the explosion of new species, and many other things. So we know already, for example, and just to give one illustration where there's a lot of data, we know that at the beginning of vertebrates, there was a couple of whole genome duplications where the entire genome duplicated itself. Extremely rare event. It's happened a few times in the history of life. And that also was a major change in evolutionary. It wasn't any longer gradual or continuous. And that's the basis of the paper that I mentioned. In fact, the title of the paper is about the origin of self-replication and the continuity principle. And the whole point of the paper is that if you're going to have accurate self-replication, you must violate the continuity principle again. So now the question is, what does that mean? When we violate continuity, was Darwin right? Should we throw away evolution? I mean, that's one argument you could make. But Darwin got a lot of other stuff wrong, not surprisingly. I mean, he lived before we knew anything about much. So what you have to do is you have to change the theory to admit mechanisms for these, what's called saltational events. In other words, jumps. But we don't have any, there is no, at this point, except for the case of the whole genome duplication, but for many other cases, we don't know what that mechanism is. And by keeping those gaps, as you could call it, or jumps or saltations, these non-discontinuity events in the science, that does open the door to a miraculous event, because we know that miraculous events are exactly that. They are jumps. They are one-time events like the biosymbiosis, right? That's a one-time event. Well, maybe it happened twice, once with plants, once with animals, but extremely rare. And so when, for example, just go off a little bit when people tell me, well, Jesus could not have risen from the dead because people don't arise from the dead. And I say, yes, that's why it was a miracle. And it only happened once. Okay.
Sure,
So
yeah.
we're in agreement. And that, we have to open as scientists, aside from being Christian, but as scientists, I think we have to be open to this idea of discontinuity, of gaps. And the endosymbiosis to me is, an example of that. It may have happened, but it may have happened through a miraculous intervention.
Okay, I understand. Of course, that would fill the gap. But then you will open other questions. Where did God intervene as well in the history of life? And if you see the even older creationists will have this problem, because if God did not create everything in 6 days, in 6 literal days, as it is mentioned in Genesis, and if you take that as long periods of time, that would mean that actually God did create in a subsequential manner, but with huge intervals in between.
Mm-hmm.
And I personally am not convinced that this hypothesis actually is what happened, because I don't see how you can transform the days described in Genesis in long periods of time.
So you're actually a young Earth creationist.
Yeah, I started as an old Earth creationist because my wife's father was a geologist. He was a Christian geologist, and he was an old Earth creationist.
Right.
And we had some conversations back then. And also there were some actually good science websites, like Reason and Science, I think, something like that, which is proposing old Earth. And I was reading these websites, and that was convincing to me that actually it could be an interpretation.
Reasons to believe, you mean?
Yeah.
Hugh Ross, yeah.
No, not Hugh Ross. It was another website. Anyway, that was quite influential to me. But then I started to investigate further, and I started to have some doubts in regards of if actually we can make that interpretation that days in Genesis mean long periods of time. And then for some years, I remained an agnostic in regards of the issue and tried to investigate that further. And then I came closer to young Earth also, because I've not seen how you can make an interpretation of Genesis as long periods of time. And I think that if God made everything, then he knows how he made it. And if he said that he made it in 6 literal days, then why not take that seriously? I mean, he is the creator of life. And if the Bible is his inspired word, why should we doubt about it, But of course, there are difficulties like the starlight problem, like the dating methods, which all give 4 billion years of the age of the Earth. And there are other problems which I think have no solution. But on the other hand, the old Earth hypothesis also has its problem, like the receding moon, like the electromagnetic shield around the Earth, and several other things which also put the old Earth hypothesis in question. So it is a complex issue, and you have to evaluate many different fields. And of many, I have no knowledge, like dating methods and geology. I have no knowledge in regards of this, so that creates difficulties. But 2 points which I regard as very, very compelling is that soft tissue was found in fossils, and I can hardly believe there are even science papers which claim that they have found soft tissues in the Cambrian explosion in animals which lived in the Cambrian 500 million years ago. So how do you justify that? And this year, I re-evaluated the claim that they did find Noah's Ark at Mount Ararat, which is something that was in the news 10 years ago in 2010. And then I discarded it as well because the claim came up that it was a forgery, that some people, the locals, they put some wooden planks on the mountain and they sold the idea to Chinese, which were gullible and thought that it was the real ark, but actually it wasn't. And then this year I met someone on Facebook, which is Philip Williams, and I have an interview at my channel. And in 2014, he went there and made 2 videos of the location. And it is very compelling evidence that it is actually a remaining of a huge ship up there on Mount Ararat. And I've investigated that for over a month. And I also talked personally with some people which brought that story up, that it was a fake. I talked to Don Patton on the phone and so forth. And he wasn't convincing. He was unable to convince me that it is actually a forgery. And now there is an expedition underway to make a scientific investigation of this wood up there on the mountain. So these 2 things, that finding, which points to the fact of Noah's Ark, of the global flood, and the soft tissues found in fossils, these 2 things, they have given me much more strength into the hypothesis that actually it could be true that the Earth is only 4,500 years old, something like that. But I'm open to new evidence. I mean, I don't think it is essential to my view as a Christian, but if evidence makes it stronger again that eventually the old Earth hypothesis is a better explanation, I'm fine to go back to that one, But for some years now, I'm holding that view that probably the Earth is young, actually.
Mm-hmm. Well, I agree with one thing. I don't think it's critical. I have had some debates with a few young Earth creationists, including Kent Hovind. And I decided not to do it anymore because it's not my mission to convince young Earth creationists that the Earth is old and evolution is true. I don't care about that very much. My mission is more related to explaining to especially young Christians and other Christians that you don't have to reject science to be a Christian, that science and Christian are perfectly compatible. That's why I wrote my book. I don't think I mentioned it, but I'm going to mention it now. This is a book that was published last year, The Works of His Hands, and its subtitle is A Scientist's Journey from Atheism to Faith, because I spent most of my life as an atheist. I was raised in an atheist family. I was an atheist and then eventually an agnostic, and I was baptized 7 years ago in my 60s. So
wow, that's rare.
It's very rare, and that's why I wrote the book. And it's been a lot of people have liked it. And I explained that actually it was science that originally, being a professional scientist and having studied biology and chemistry as I did, that put cracks in my original atheist worldview, and realized that it wasn't sustainable. It wasn't real. It wasn't right. And so I was able to get away from that. And then eventually, through other things, I came to Christian faith.
So what triggered actually your change of position?
Well, again, that's in the book. That's the whole book is about it, but I'll summarize it quickly.
Yeah.
The first thing was when I started learning about quantum mechanics in college. As a chemist, I was a chemistry major, and you have to learn how to solve the Schrodinger equation. And when I started looking into what that was all about, it didn't make any sense at all. I had been learning about a deterministic, very materialistic universe, clockwork. And of course, quantum mechanics throws that all out the window. And, now you have the observer effect, and you have, all the odd things about real reality, quantum reality. And I started thinking, well, if that's true, then a lot of things could be true, right? And later, people have said to me, well, how can you believe in in Jesus as both God and human. And I said, have you ever heard of an electron? It's both a particle and a wave. So, it doesn't completely shock me that that there are things that we don't understand. So that was the first real blow that I had. But then I began graduate school. I started learning biochemistry. I mean, deep biochemistry. And I don't know. There are plenty of biochemist atheists. In fact, I guess most of them are atheists. But I just find the incredible complexity.
Which astonishes me, because
yeah, me too.
I mean, I studied biochemistry for years, and the more I study, the more I am astonished about the design which I see in biochemistry, and the evidence which is, in my understanding, just mind-blowing. And I mean, I have conversations with atheists basically on a daily basis, and it really doesn't go in my head how someone can actually understand what goes on inside of the cell and nonetheless remain an atheist. That is something that I can't understand.
Well, I guess I can understand that, but what I don't understand is, okay, maybe you I mean, I had a very strong commitment to atheism. It was part of my worldview. And I just was uncomfortable because, as you said, the clear evidence of design of incredible complexity just leaves you wondering. And what I find is that atheists who are biologists or biochemists or biologists or even chemists, what they have answers, and you'll find those answers in Richard Dawkins' books. It's kind of hand-waving answers. And what they do is they make up stories, many times related to evolution, which sometimes could be true. But
I mean, one thing is when you understand biochemistry on a superficial level, but once you are starting actually to go deeper into it, then I think that things become very complicated to remain an atheist, because materialistic mechanisms simply, in my understanding, they do not withstand scrutiny, because in special, the selection problem is a huge problem. How do you select the right molecules to kickstart life? The 4 basic building blocks, there was no selection mechanism there.
Exactly.
And secondly, Steve Benner wrote a very good science paper in 2012, the paradoxes of life, and he pointed out that molecules, they devolve and become asphalts. They do not complexify by themselves. So that's another huge problem. Yeah. And the third is the information problem. Right. How do you get the information? Then you have the energy problem. How do you get the energy to kickstart it? The whole thing. I mean, these are.
I'll tell you what some atheists do. You mentioned the information problem. And that has to do with, how did we end up with the genetic code at all? How do we have DNA that actually carries information? When, if you think about it, there isn't anything else in the universe outside of life, again, that carries information of that type.
Yes, and I mean,
go ahead, sorry.
I mean, one problem is the information, but the other problem is also the assignment of meaning from carbons to amino acids, which is also something which I say assignment of meaning is only something which intelligence does. We do not have an example of where unguided events have that capability in principle, actually, to assign meaning, to assign something which means something else. And this is, in my understanding, a conceptual problem. It is, unguided mechanisms simply are not the adequate explanation, and we know only of intelligence which is capable of doing that.
Well, the answer to that, which is very interesting, when you ask an atheist, not a scientist atheist, but just a regular atheist, about that question, a new answer has come up. It's only been around a few years, which is that DNA is not really a code.
Oh, yes, I hear that all the time.
You hear that? Okay.
Yeah, all the time.
Rationality Rules, Stephen Woodford, who is a British atheist.
Yeah.
In 2017, he made a video claiming that DNA is not a code. And I made a response video to that,
Mm-hmm.
showing his mistake. He gets confused with the terminology. He thinks sequence is code. It's completely confusing.
Yeah.
But that meme has spread throughout the atheist community. And every time I'm on social media, especially Twitter, excuse me, I will see people saying, and these are not scientists, but they'll be saying things. Well, DNA is a metaphor. It's a metaphor for a code. It's not a real code.
Yeah,
that's complete nonsense. It's like a real complete code, and every scientist knows that. It's in every textbook.
Yeah.
I had one debate on the old Non Sequitur show with Herman Mays, who's a biologist. And,
I've debated him several times on Facebook.
And the debate was, is DNA a code? And I made my opening statement, and Herman made his opening statement, and he basically agreed with me. I mean, you can't be a scientist and deny that DNA is actually a code. So but that's what people do when they're stuck with a problem that they can't answer. They make one up, and everybody does that. I mean, you do it, I do it. We all do it to some extent. But the idea that I would like to convey to Christians, to people who are worried about, is science telling us that Christianity is wrong? Everything can be explained, quote unquote, explained by natural laws. The answer is, well, look at real science and look at it carefully, and you won't find any arguments against God. There is nothing that contradicts the actions, existence, or presence of God and of Jesus Christ in your life. I mean, those things are not connected to scientific denial.
Well, that is a standard question, which I always ask to atheists. How can we explain our existence without a creator?
Mm-hmm.
And there are some which say, well, the gap is shrinking, and the more science advances, the more it will there will be natural explanations. But my reply to that is, I don't know any compelling explanation whatsoever in regards of everything related to origins.
You're right. And in fact, the idea that the gap is shrinking is wrong. It's the opposite.
Yeah.
The more we learn in the last, say, several decades, the wider the gap is getting.
Yeah.
Now we have a question that we never had before. What is dark energy? What is dark matter?
Yeah.
Why are the cosmological constants so finely tuned? Talk about life, abiogenesis. How did life begin? How do you get over the fact that RNA cannot self-replicate? That you have to have accuracy. All the stuff that's in my paper, and it's also in the book. I mean, science is not closing these gaps. More gaps are coming, and some questions will be answered. They always have been, and they will be answered, but not all of them.
So, Sy, would you say that the laws of physics, are they only described, or are they prescriptive? Are they prescribed?
Well, clearly, as a believer in a creator who created the laws of physics, I would say that they are prescribed.
Yeah.
But you're right. That's you hear that all the time. No, they're just descriptions of reality. What does that mean? I mean, I don't even know what that means.
Yeah.
But I think it's important. Having said all of this, it's very important to understand that we can understand the natural world through scientific investigation up to a point. And that point keeps changing. Okay. So in the 19th century, if we were in the 19th century, we obviously wouldn't be talking online, but let's say we met, in a cafe, and I said to you, "Oh, Tangela, you want to know something? I have this idea that there's this electron, and somewhere out in space, there's another electron that's tightly associated with it." You'd say to me, "Well, you're crazy. Okay, you're nuts. That can't happen." Right? Well, it's true. It does happen. So in the 19th century, that would have been considered way out, crazy, supernatural, magic, stupid, right? Now it's reality.
You are talking about entanglement, correct?
Entanglement, right? Exactly.
Yeah.
But not only. I mean, there's so many other things. That electrons only go through the two slits when you watch them. I mean, the observer.
Yeah.
You can just say so many things. If I started talking about gene regulation to somebody in the 19th century, their mouths would drop open and they'd say, "This is fantasy." I mean, the way different genes regulate the expression of other genes is just beyond even the word complexity. It's just incredible.
But that is another point, Sy, which I had recently some conversation about it. Is Boolean logic, is that just something that we observe, or are genes actually literally regulated by logic gates?
That's a good question. I don't know the answer, because actually logic gates is almost too simple. It's probably beyond that. A very simple picture map of a gene regulatory pathway will have a few logic gates, but that's not how it works in biology. It's much worse than that. So, my point is that what we consider hard, definite science and what we consider, supernatural or crazy stuff or whatever, that border keeps changing. That goalpost keeps moving back. I mean, and that's going to keep happening. And at some point, I believe somebody's going to have to say, okay, guys, you know what? At this point, I think we have to admit it. It's God. When will that happen? I don't know, but I think it's going to happen.
So let's move to evolution, Sy. So you believe that do you believe that the standard explanations in regards of biodiversity, which is the gene-centric view, that still does withstand scrutiny, or what is your view on that? How do we explain biodiversity today? What is the best explanation?
The biocentric view has come under attack.
The gene-centric view.
The gene-centric view, sorry.
Yes.
The gene-centric view has come under attack strongly. It started slowly, and it's interesting because I was originally a gene-centric person. I worked on DNA a lot. I love DNA, I love genes, I love the idea. When I read The Selfish Gene, I thought it was the best thing ever written. I was also still an atheist. But that's come under scientific attack from many fronts. And it's now clear that evolutionary pathways are far more complex than the simple idea of, okay, you have a point mutation in a structural gene, the expression changes, it makes a phenotype that's better for the organism in a particular environment, you get natural selection, boom, evolution. That's sometimes called neo-Darwinian. It's really, it's so much more complicated than that. You have neutral evolution, which has no selection, which is very important. You have the Simon Conway Morris's ideas of convergence, where you have the same structures being produced in completely unrelated sets of organisms. You have punctuated equilibrium from Stephen Jay Gould, which says no, it's not a steady continuous thing, and that's just from the evidence. He was not a theist; he was an atheist. But he looked at the data and he said, yeah, sometimes nothing happens, and then boom, you get this big change. And then you have you have the whole idea of genetic engineering from James Shapiro. You have Kevin Leyland with a niche construction, which is a two-way street between the environment and the genome.
Yes.
I mean, I go on and on. In fact, I've written papers about this on the extended evolutionary synthesis. So, yeah, it's gene centrism is struggling because there's epigenetics. There's even the question, how do you define a gene? Okay, if you have a gene which mutates, then its sequence isn't exactly the same anymore. Is that a different gene or the same gene? Okay. This is why it's very hard, for example, to make species out of bacteria, because their genome is so fluid that if you compare genomes, E. coli today and E. coli, 50 years ago, they're not going to be the same. Okay, some will be the same. There are different strains, and as a strain of species, I mean, it gets, the thing about
you have horizontal gene transfer.
Oh yeah, sure, exactly.
And of course that scrambles the whole thing.
Scrambles everything from one organ, and even eukaryotes can do that. So, I mean, I didn't go through the whole list, but there's gene duplications, et cetera. There's this whole list. And so what we're left with is the recognition that the way we think, with things in nice, tidy corners, okay, we are engineers, basically. We meaning the human race. That's not how God thinks. Okay. God is not, and by the way, this is why I don't like the word intelligent design. Because I don't think God is an intelligent designer. He's way beyond that.
He's a super intelligent designer.
I call it divine design. That's the word I prefer.
That's a nice description, I would say. Yes, mm-hmm. a super intelligent divine designer.
Exactly.
Yeah. Sy, I have made some extensive research in regards of the mechanisms which explain organismal complexity. And I would like to put it here on the screen what my view is today. I think that it is much, you said the keyword, which is epigenetic information. Mm-hmm. And I agree with you that I think a large part of the information which defines how organisms are built is not stored in genes, but in epigenetic languages.
True.
And I have put here a list of 19 different mechanisms. I've put the link here on the side chat. I'll put it also in the private chat. And after our conversation, you can have a look on it, and then eventually.
Yeah, I'm looking now. It looks very good. Yeah.
Yeah.
And, absolutely.
And what is interesting, I've collected 29 different epigenetic codes so far. Okay. So all these codes here, they are also involved in making organisms.
Yeah.
So
I see what you're saying. I think I understand this. So you're talking about gene regulatory network codes, right? Is that what this is?
Yeah, adhesion code.
Because I see you have the Hox code for development, transcriptional, cis-regulatory code, right? Okay. It's kind of small for my eyes. Okay, yeah. I mean, right. This is straight standard science.
Yeah. So in my understanding, it is actually not just genes which construct organisms, but it is an entire integrated system on a systems level of which orchestrates the whole thing in a joint venture of very different languages which actually work together in a very, very complex manner, which science is far yet to actually unravel and understand. But that brings us actually back to a designer which had to preload the information at the beginning and then permit that organisms adapt to the environment, but only in a limited manner, and not as evolution makes believe that we start from just one less universal common ancestor, and then from there all different life forms evolved.
Well, the LUCA is not the original life form. That's obvious. It couldn't be because it's so complex. The only value of Luca is like the Big Bang. Okay, it's as far back as we can go.
Yeah, they also mention the first universal common ancestor, but tend to explain what it would be. That would be also very difficult because, I mean, as I said previously, in order to have a minimal free living cell which can self-replicate and which can synthesize the basic building blocks of life and has all the metabolic networks and information and energy and so forth, as you said, it's already extremely complex.
Right.
But if you do not have just one import channel on the surface, on the membrane, which imports iron, for example, or sulfur, then you have not the building blocks for making several life essential proteins which use cofactors which use iron sulfur centers. So I think that this brings you to a big problem in attempting to explain that there was a previous first common ancestor which had a smaller gene set and then started to self-replicate. I mean, only prokaryotes, they require about 30 proteins to start to make DNA replication, and you are just replicating DNA, but you have to replicate all the rest as well.
Right, and the point is, and that's my point, and that's why I wrote this paper, because you have to replicate the entire phenotype of the cell in order to get evolution started. So how do you do that? And the way you do that, you can't do it with RNA because the RNA world can't do everything. But so how did Luca and everybody else after that do it? They do it by making proteins, making all the enzymes. It's the enzymes that give the phenotype to a cell. But how do you make a protein? And here's where we get it. I'm going to touch into chemistry a little, so don't run away. But the fact is that the only thing that can be replicated is a nucleotide polymer like DNA or RNA, because of the base pairing of the A and T and C and G. That proteins can't be replicated directly.
Yeah, and you need proteins to make DNA replication.
Exactly.
And that brings us to a catch-22 problem.
Exactly. And so the question then is, okay, so you have the DNA which carries the information and gets replicated, but now you have to translate. You have to come up with a translation system, which is what it's called, protein synthesis.
Yeah, the transcription and translation, and you also require already complex proteins to do so.
Yeah. So the issue is, how do you translate nucleotide chemistry to protein chemistry? Because those are two completely different types of chemical molecules, and they don't. They will bind. Some will bind to each other, but they don't. You can't do that. You need an incredibly complex translation system, which includes transfer RNA and ribosomes, and of course these incredible enzymes called aminoacyl tRNA synthetase, which actually translates the RNA and the amino acid to each other, and that's where the genetic code originates.
Yes.
But how do you get that? And that has to work well. How do you get that working well without evolution?
Well, I've investigated that, and you have 13 different translation error check and repair mechanisms in the ribosome.
Right.
And in order to have sufficient accuracy in translation.
Right.
And amino-acyl tRNA synthetase, they have internal signaling information transmission where they check if the right amino acid is being added to the
Good for you. There are very few people who know that. That's I even know some biologists who are not aware of that. So I give you a great praise for knowing that fact. That's an incredibly important fact. They have an editing site in the protein, in the enzyme.
Yes, and also the ribosome. I mean, I've studied significantly the translation process, and I mean, and you need about 200 different assembly and chaperones to assemble the ribosome, and about 75 different cofactors for the assembly. And you require that all prebiotically in order to start life, because translation is a life essential process. And you have no evolution to explain it.
Right. So let me bring up something, which is I started saying that I raised this issue to a fellow Christian, real origin of life researcher. And he did not agree with me. And he said, well, it's possible that in early life, there were other mechanisms, other systems that worked. Okay, fine. That's not an answer, but it's the best he could do. That's why I started working on this project that resulted in this paper, because what I have done in this paper is I said, okay, forget all the details of the biochemistry. Forget it. We don't know what the details were. Is it theoretically possible, no matter what the mechanism is, for a cell with a very low replication accuracy to survive and evolve? And how high does that replication accuracy have to be? And I did it through a very simple statistical model, which I developed into a computer program, et cetera. And the results were very surprising and very striking. And that's in the paper. And basically, the conclusion is, this is discontinuity. If you start out with a cell that has less than 65% accuracy in replication, nothing can happen. The cells can't survive. Certainly they don't evolve. Done. So how does a cell get to 65% accuracy? That's not bad. Okay. That's a pretty accurate attempt. But it's not enough. You have to get above that. So if you have a cell that's 50% accurate in its replication, in its whatever the mechanism is, it's doomed. So
and you have error check and repair mechanisms all along the pathway.
Exactly.
It starts with DNA, it starts with RNA polymerase, it starts with the ribosome. There is even error check. There is the chaperones after the protein comes out of the ribosome, and then it is also checked. And if it's not okay, then it is discarded and recycled. Amino acids are recycled. And even if you have a protein which comes out perfectly from the ribosome, you need still information to know where it has to be placed in the cell. And
right.
You have the sequence, you have the signal recognition particle, and you require to tag the protein that it can be transported to the right place. And you need all this information.
Right. So
you need all this information right from the beginning.
That's right. And the point is that you can make the argument that some of these perfections came later with evolution, but the question is, if you don't have evolution, how did you do that? And the reality is, I mean, you know what the accuracy is now of DNA replication and translation. Just tell the audience, DNA replication is accurate at 99.99999%.
Yeah, I think it is in 10 billion nucleotides you have an error, something like that.
It's 0.00001% error. I don't, and the same thing is true for the translation system. It's a little less accurate, but it's 99.9999% accurate.
Yeah.
So that if you just take ribosome and RNA and all the ingredients and mush them together, you may get 40 to 50% accuracy. I mean, you need all that other stuff that you mentioned to have anything close to 90, never mind 99.999. And that's true in all modern life. So, there's a big gap to fill. And I think this paper, I hope people read it and understand the point of it. I'm going to be writing another paper that's less technical, that I'll put into a journal, not necessarily for scientists. But I think that there's not much answer to this, because this is making no assumptions whatsoever. It's just assuming you could take any system you want. The same effect will occur. And this is just based on statistics. So that's where we are.
Yes, I have here. That's the combined. There are 3 steps in regards of DNA error check and repair, and it's 1 in 10 billion. One error in 10 billion.
Right.
That's just in regards of DNA error checking. I mean, that is absolutely astonishing. And
yeah, 1 in 10 to the 9th. That's right. That's the error rate.
And
that's true. And by the way, that's true across all of life, which is amazing. So we have the same error rate as a worm.
Yeah, I mean, how do you get that? And I think another huge problem, Sy, is that we do not know of any life form which does not use DNA.
That's right.
And DNA, I think that the transition from RNA to DNA is enormously complex.
Nobody has a clue.
And the ribonucleotide reductase enzymes, I don't know if you have studied those. They are the core enzyme which makes the transformation in the transition from RNA to DNA, and it reduces I think it's at the 3' position or something. It reduces 1 hydrogen, and it just remains oxygen.
Right.
Yeah, and I mean, if you study that enzyme, it is enormously complex.
Yes.
And you need that to make DNA. And how do you get to that enzyme prebiotically if there was no evolution yet? Because you need DNA to have evolution.
Yeah, I mean, there are so many questions of that. By the way, RNA world is now starting to fall into scientific questioning, just like gene syndrome.
Science fiction.
Well, it was very popular for a very long time when it was discovered about ribozymes, that they have enzymatic activity, and the ligase, the enzyme that joins the 2 amino acids together is a ribozyme in the ribosome. So that I had some fierce arguments with various people on the BioLogo site.
Yes, it is a ribozyme, and it is interesting, Sy, that in order to have a correct protein folding, that there is a twist at the moment that the 2 amino acids, they are bonded together. During that process, you need actually just a 45-degree twist. And they have figured out that in the ribosome, the twist is 180 degrees. It is more than just what is necessary. And they have tried to find out why that is so. Why does the twist be 180 degrees and not just 45 degrees? And they have they did test, and they did figure out that this helps in the correct folding of the protein when it comes out of the ribosome. And I think this is astonishing.
That is astonishing. I didn't know that.
Yeah, I can give you afterwards the link of my studies on the ribosome. And already when the protein is starting to get out in the channel, in the exit channel of the ribosome, it is also there error checked. And if there is some kind of error in the sequence or in the folding, then there is going on signaling between the tunnel, the exit tunnel, and the assembly site, and it stops. The ribosome is able to stop the building on the fly and discards the sequence. I mean, this is a technology of the highest order.
It also gets tagged for degradation by proteases very quickly. So
yes. So
I mean, it's an amazing field. I mean, I'm very happy that I went into biochemistry, but it moves so quickly that I can't follow everything that happens. But it's just fascinating, all of this material, and and it just leads you to a sense of worship. I used a quote in my book, which I use frequently. It just came to me one day while I was writing. And it's sort of the theme of my book, which is that science is distilled doxology. And that's what I mean what I mean by that is what we've just been talking about. As you study how the ribosome works, you could study how one enzyme works. You could study any part of the amazing field of biology that you want to. Photosynthesis, my God.
Oh.
I mean, it involves quantum mechanics of all things.
Yeah.
I mean,
I made a video on photosynthesis at my other YouTube channel. It's called Intelligent Design Academy, and it's mind-blowing. I mean,
exactly. And this shows you that when you study science, you're praising God. That's the bottom line to me. And that's why I say it's distilled doxology, because it's pure. It's pure worship. It's pure praise of the Creator. That's to me, that's what science is.
I mean, for me, it's on a daily basis, flabbergasting and being in awe when I discover something new.
Good. That's the word.
Yeah.
All right. Listen, I'm going to have to sign off soon because I've got a couple other things I got to deal with. Sure. And but it's been a lot of fun talking. So
I'm having fun too, and I have very frequently chats, not so much at my channel, but I can give you the link at Speed's channel, and they are atheists, and I am pretty much a lonely theist guest there. And
this is standing for truth.
No, it is a Speed of Sound of Gravity. Is the name of the channel. I can
okay. No, I don't know that one.
They're most atheists, but Dimitar Kunev is frequently there and interacting too. So I will give you the link, and then eventually, if you have fun, it would be welcome that. You can a little bit balance out the forces there with me.
Okay.
And put a little bit your input in our conversations.
Okay. All right. Thanks for having me on. And you've recorded this, I presume, right?
Yes. So we are live. And
okay.
Yes, it was my pleasure to have you at my channel, and I hope that we can keep in touch and advance our conversations. And
let's do that.
Keep sharing ideas in regards of all these fascinating fields.
Yes, indeed. Okay.
Okay.
Blessings.
Okay. Thank you very much. Thanks, everyone. Bye-bye. See you next time.
Bye.